It's a pleasure to be here, and I can tell you it was 36 degrees, overcast, and raining in Seattle when I left yesterday. So coming to a place like this is glorious, so thank you very much. And it's wonderful to be here at a place like this. It's always frightening to talk after Michael talks because there's such a wonderful success story. So what I'd like to do is to talk to you about a story about how we began to recognize that there may be syphilis and what the genes are and what the phenotypes are. In the late 1970s, David Silence, who's a medical geneticist in Australia, looked at the then current classification of OI, which was basically non-existent, and it was sort of barely organized chaos. And he did a genetic experiment, which was a very simple one, which was to find all the people in the area that he could and begin to look at them with clinical criteria, radiographs, and take a family history, which was not usually done. And was able to identify 4 different groups that he could see, and they became the standard for the, until fairly recently, standard diagnostic categories of osteogenesis imperfecta, which is OI type 1, which is a mild dominant disorder with usually fairly normal stature, blue sclerosis, and no major deformity usually. OI type 2, which is a perinatal lethal form, which he decided was an autosomal recessive form, and you'll see why in just a moment. A congenital, also congenital progressive deforming variety here that was very severe at birth but got gradually better over time. These kids usually didn't walk. Most of these kids died in the first week or so after birth. And then there was the leftover group. These group usually developed over time more or less Whyte sclerosis, were usually small, had dentinogenesis imperfecta, as did this group. These guys don't have overt DI. So the idea that there were recessive forms was based on pedigrees like this. If you're a geneticist, you realize, and Bill talked about this earlier and talked about the wonders of cousin marriage, and I don't know about you, but I thought my cousin was hot. So you can understand it, you know. The problem with cousin marriage can be, if you come from the right environment, that you're at risk for having children that have recessive form, have recessive disorders. And in this family, you can see that there were 3 affected kids who had really very severe forms of osteogenesis imperfecta. In this family, David thought that they might be related, but he couldn't trace the consanguinity back. So this was a very interesting notion. Right beginning around that time, we began to recognize that the majority of people who had brittle bone disease, osteogenesis imperfecta, had mutations in collagen genes, and we could see abnormalities. This is a complex picture. The collagens, type 1 collagen is transcribed, obviously, in the nucleus, translated on the rough endoplasmic reticulum. The chains come into the rough ER lumen. There's extensive modification, and all of these little colored dots represent different modifying or chaperone-like proteins. It comes out of the ER, goes through the Golgi, and then is secreted, where there's additional modification out here. And our experience in analyzing people is that, as far as we can tell, about 95% of everybody that has OI has a mutation in one of the 2 type 1 collagen genes. So 95% have it, and that's looking at sequences in just under 3,000 people. So I think it's a reasonably robust number at this point. So given that, we and others had begun to look at the clinical features, and here is a series of kids with the lethal form of OI, and it ranges from radiographs like this that would be difficult to distinguish from some of the kids who have hypophosphatasia with virtually no skeletal mineralization, to others that are over here. And there were families in which there were recurrents that we could see, just as David had seen. Some that were like this, some that were like this. Here was one that would be potentially recessive, and others like this. But the families that were really unusual in this respect were ones like this, where there's a woman who's had affected children with apparently two unaffected guys. There are lots of ways of explaining that. You can think of most of them, but one would be that these are brothers and that that would constitute, you might have a recessive disorder on that. The other is that, in fact, this guy is also the father of this child. We know it never happens in ordinary families, but you would be surprised. And as we do genetic studies, we recognize how common things like that are. So we recognized in looking at these that, in fact, they were heterodominant. And the explanation for recurrence was awaiting a family like this one, where Bruce Blumberg from San Francisco provided us with a family in which this guy had two affected children. The striking thing, the unusual part of the story, was after this child was born, as he was walking out of the room with a genetic counselor, he said, this is really kind of unusual. My girlfriend just had a child with this a little while ago. So genetic counselors are wonderful creatures in the environment. They can hear everything. And the explanation was that the dad is actually mosaic for that mutation. So he has a small number of cells. The mutation occurred very early in embryogenesis. It's transmitted into the germline. It's seen at very, very low levels in somatic cells, insufficient to give him a phenotype. And this became the explanation that we understood. Subsequently, we've recognized that for those families in which there is a first affected child, so if we look at the first one in the family, about somewhere between 1 in 16 and 1 in 20 of those families has a mosaic parent. And so in general, mosaicism for this outruns recessive disorders by a substantial amount, depending on the population in which you look. So that actually, that put the kibosh on the search for recessive forms of OI for about 15 years. However, what happened was that, in the course of going through things and looking, everybody recognized that they had families sitting in their clinics, in their freezers, in their wherever it was that they collected families, which didn't have mutations in type 1 collagen and looked like they might be different. And they began to see families in which there was consanguinity. So there was a rush through now to begin and identify. And there were two different strategies to develop new types of OI that had clinically differentiated from this 1 through 4 group. And the first was really a clinical presentation. And this, on the basis of that, OI type 5, OI type 6, and OI type 7. 5 was probably dominant, has proved to be. 6 was probably recessive, has proved to be. And 7 was clearly recessive. They had no collagen protein abnormality. Their sequences of the type 1 collagen genes were different. Sometimes they were distinctive populations, and sometimes consanguinity was the draw to find the genes. The next thing that happened, the development of strategies, and all of this has happened primarily since about 2006 or 2007. So a very rapid uptake of the look for these new things. The others were the use of candidate genes based on studies that people had done, and the other was what Bill talked about, which is exome sequencing, which has become a very powerful tool for finding ones that are missing. So OI5 is represented by this very dramatic hypertrophic callus formation that can occur almost anywhere. It can occur after injury. But this is a remarkably variable phenotype in families, with some people who have essentially no manifestations and other people who are quite short with very dramatic abnormalities. It's a dominant disorder. Everybody who has this has exactly the same mutation. It's a very unusual, very unexpected mutation. Tied upstream from the start code of the translation start codon, that creates a new translation start site that actually has a better surrounding sequence for ribosome recognition, better Kozak sequence. And so there's the addition of 5 amino acids on this protein, which is a transmembrane protein. And at this point, I think that there's no clear evidence of what this protein or how this protein works. But very dramatic, and it's called, oh God, interferon, thank you, interferon transmembrane protein 5, except that it's not responsive to interferon currently. So So, like many of us, it's got the wrong name. You know, it really liked its cousin. That's all I guess. So, Whyte type 6 is characterized. These kids look normal at birth, which is very striking, and they develop their bone disease as they go on. And by a few years of age, they begin to get significant scoliosis. They get very marked bowing. And they have mutations in SERPINF1. They have biallelic mutations. And again, the function of this protein has been studied a lot, but the function in bone disease is not clear. It's a collagen binding protein, but how it actually affects the phenotype is not clear. The real start so those were very recent. Those are all in the last couple of years. The real start for the analysis of recessive disorders came from the group in Montreal, where they were studying the First Nations group in the area and recognized that there was a phenotype with what they called rhizomelic shortening and bowing, and they called this OI type 7. At the time, it had a distinctive histology that they found here. And they did linkage studies in the families and showed that it was on the short arm, that apparently linked to the short arm of chromosome 3 in this region. And you can see that there is sibling recurrence. And with the right pedigrees, you can actually trace people back to a common ancestor in many of these groups. So it was certainly consistent with a small population group and the likelihood of having the same allele throughout. At the same time, Roy Morello, who was in Italy, identified collagen. He was interested in cartilage development, identified a protein that he called CRTAP, which was supposed to be a cartilage-specific protein. When it was knocked out in mice, which he did with Brendan Lee's group, he found that the mice grew more slowly. They had abnormal bones. You can see that they mineralized less well over here. Here's the normal. And this, I wouldn't tell you this story unless this gene mapped to exactly the right place. And in fact, it mapped into the region that had been seen in these families. And they went back here and find very difficult to identify mutation. They missed it the first time through because it involved the creation of a new splice donor site in the middle of a fairly large intron. So this is a new exon in the gene, and it occurs because it's got a single nucleotide change here that creates the new donor site. So by ordinary sequencing of the exons and everything, they absolutely missed it. They actually sequenced through there. Genomically, you didn't see it, and didn't see it until they actually looked at the cDNA, when they could find that about 10% of the product, about 90% included this, and 10% was spliced normally, which accounted for the relatively mild disorder. And with a premature termination codon in both alleles, which inactivated, which made the RNA unstable, there was a lethal phenotype that was seen over here. So this was the start of the new run on recessive disorders. The thing that was very interesting that was done at this point was that Hans-Peter did a very interesting experiment, which was to look at proteins that will bind to collagens and recognize that you can take those proteins and separate them first by density centrifugation and then by polyacrylamide gel electrophoresis. And the advantage of doing this is an interesting one, and that is that proteins that may have different sizes, like this one, and this one, and this one, migrate to the same position on density centrifugation, and it means that they bind to each other so that they form a complex. So it was a way of finding things. So here was CRTAP, right here, and it bound to prolyl 3-hydroxylase and also bound to cyclophilin B. And this led to the next phase of things. So Joan Marini's group was first out of the box to publish the P3H1 mutations, and then a bunch of other people identified cyclophilin B mutations. So these are, they form a complex. The complex is strongest between CRTAP and prolyl 3-hydroxylase. We've never been able to show that cyclophilin B is actually in the complex. Hans-Peter Bockinger insists that it is, but it's very hard to show. And the phenotypic range, if you have low levels of any of these things, of CRTAP or P3H1, You can have moderate phenotypes, and if it's absent, they're lethal. So for why do mutations in LEP1 and CRTAP, where they're null, have similar outcomes? You can see these X-rays are very difficult to distinguish from those with the perinatal form of OI that's due to collagen mutations, except that they tend to have slightly less bad ribs. But certainly the lower extremities look very much like that. It turns out that these proteins bind to each other and that they're not stable without the other protein present. And so if you have a mutation in LEPR1 and you have a null, you see no P3H1, but you also see no CRTAP. Here are the controls, you see them both. Here are CRTAP nulls, and you don't see either one. So these proteins must meet each other very early on in the process of being synthesized to be stable. And as you can see, cyclophilin B may get into that complex somewhere, but it doesn't necessarily mean that it's dependent on its stability. Mutations in cyclophilin B, the PPIB gene, range perinatal lethal, looking just like the standard perinatal lethal one, to mutations that are here. This is one null and one missense mutation. These are two missense mutations. And we think that in addition to being active involving the triple helix, so the prolyl cis-trans isomerase activity of that gene looks like it's responsible in part for making the very proline-rich collagen fold properly. We think that it also has an effect on chain association. And all the experiments that are done prior to this really can't distinguish those. We think that there is clearly an effect, and there's a delayed chain association because we think the carboxy-terminal propeptide of type 1 collagen molecules, which have to fold correctly to get the molecules together, don't fold correctly in the absence of this protein. So at that point, there were other candidates on that thing, and people came looking. Ulrike Schwartz in our group found a mutation in SERPINH1. This was a gene that encodes HSP47, a protein that had been studied extensively. The knockout in mice is an embryonic lethal very early in embryogenesis because they don't make collagens effectively. So what you can see is that this looks like a moderately severe form of OI. These are normal X-rays over here. You can see rib abnormalities. You can see bony abnormalities. You can see the effect of bisphosphonate treatment. This is a child that died at about 3.5 from a respiratory disease. It's not clear that it was related to the bone phenotype. The interesting thing was that at the time that we did this, oh, what's missing? It fell out of here. Just before we did this, a mutation was identified in dachshunds in the same gene. And I thought that this was a particularly unfair bit of nature getting around. I mean, if you're going to do something, don't do it to a dachshund who's already, way down there. I mean, maybe not having such long bones means that they do better, that they don't have such long angles. But what happened is that the striking thing, I don't think it's, oh, there it is. Mutation in people is right there. It's a leucine to proline at that position. And the dachshund mutation is at the other end of the molecule. It's a leucine to proline. So this molecule binds as a dimer to collagens. It's held to collagens by an arginine to aspartic acid bridge in here. And it's responsible for the final phases of putting this molecule, kind of squeezing it together. It's sort of like, your butler. You don't have butlers, but in the old days, your butler would make sure you didn't go out with your hair uncombed and things of that sort. And that's very much what this protein does. And so it anchors at arginine sites and then gets the molecule back into place. And the interesting thing about this was when this crystal structure of this association was looked at, it turns out that the leucine at 78 and the leucine at 326 are adjacent. And it looks like they're part of the molecule that actually holds them together and stabilizes them. So it was an answer. I mean, why would something there and something there, both of which are missense mutations, do have very much the same kind of phenotype? Here's why. It's a beautiful picture, a beautiful answer. And we could see that if we took molecules from proteins, if we took the collagens from these proteins, made by cells from this child, we could see that there was much more degraded protein. So the SERPINH1 HSP47 seems to identify arginine-rich regions along the molecule. Normally, it squeezes those and gets them into the right configuration. And then it allows these proteins to leave the Golgi. If it doesn't, we get, I'm not going to go through this, but you get accumulation of protein. It comes out of the ER very quickly. So HSP47 seems to be a guardian of keeping the unfolded protein in the rough ER to allow it to fold, and instead it gets into the Golgi very quickly. So the next round, and this disease was described, or the name was given to it early on as Brooks syndrome, and these are contractures and fractures. So this is Brooks' original patient, published in 1897, I think, here, and you can see very marked fractures. The striking thing about his patient was that those fractures weren't present at birth, and they developed over time and probably developed with fractures that occurred in relatively poor orthopedic treatment. In contrast, there are kids who do have fractures and they have contractures. He's not just sitting there. He can't get his knees straight, so he has contractures. And they have significant bone disease. And Ruud Bank and colleagues in Holland recognized that there's something wrong with collagen cross-linking. Collagen crossing occurs because there are lysyl groups within the triple helical domain that are hydroxylated by lysyl hydroxylase, and also in this little extension called the telopeptide, which are hydroxylated. And they're actually substrates of different enzymes. PLOD1 encodes lysyl hydroxylase 1 that encodes these lysyl residues. PLOD2 hydroxylates this one preferentially and this one preferentially. And without them, mutations give rise to Brooks syndrome on the one hand, but interestingly, mutations in this gene, FKBP10, which encodes FKBP65, don't ask, give rise to an overlapping phenotype that's almost identical. So they share an abnormality. It's been difficult to show, but they seem to interact both with HSP47, so that these molecules, as they're coming out from cells, are a little less stable, and also with lysyl hydroxylase 2. And this was a paper from the early group. These were the early FKBP10 mutations. And here is one. So this is from a family in Samoa, where the carrier frequency for a specific mutation is probably about somewhere between 1 in 80 and 1 in 100 people. And we identified with Tim Cundy there and other places, 10 families, didn't know each other, all had the same mutation. Speak about cousins, you know? So, and they have a characteristic clinical picture which you can distinguish. They have early onset contractures. So this little boy looks a lot like the one that you saw. They developed acetabular protrusio, which can be quite severe. They develop scoliosis by teenage years. And here you see again the acetabular protrusio. And the defects are, the initial ones were identified by Debbie Krakau's group, and from a family in Turkey, and again with multiple families that have the same thing. And then here are more mutations, and a bunch of groups have identified these. But the defect is actually in cross-linking and producing the hydroxylation of that group. Now there are a few others, and again I'll tell you what they are. BMP1, we talked about the relationship between BMP2, which actually does things in terms of activation and stimulation. BMP1 is a protease. It's responsible for cleavage of the carboxyl terminal end of type 1 procollagens from the end and for some other proteins. And there's one family described. And if you get the sense that you can't tell the difference among these kids, I think you're right. I don't think I can distinguish these clinical pictures, these recessive ones, easily, one from another, until a certain point in them. So the reason for that is that these all come out of the OI3, OI4 group that have been kept in people's clinics and refrigerators and freezers, and they just sort of pull these guys out and study them, and they all have overlapping phenotypes. So BMP1 is one example. and there's a defect in the cleavage of the carboxy terminal end. Dominant mutations that affect the substrates here give rise to a dense bone disease, interestingly, without skeletal deformity. The DNA binding protein, ostrix, encoded by SP7, was identified in one family. And again, the clinical picture looks very similar. It creates a truncated protein that there is no functional studies on this. We just identified a second one, which is a compound heterozygote in this gene, so we don't know. Very interesting gene found in Saudi Arabia. Not found, but the mutations were found. It's a 20 kilobase deletion and with a 12 base pair insertion that takes out exon 4. It creates a frame shift in an unstable protein. This protein was identified first in the mid-2000s in Yazawa's paper in Nature. Sees this as a membrane, as a transmembrane protein with a partial transmembrane thing here that's involved in calcium homeostasis in the cell and in the ER. And the final one is the newest one. So Shauna Pyatt, who's supported by the OI Foundation, where is Tracy, with thanks to the OI Foundation, using exome sequencing, identified Wnt1, first in this family and then subsequently in several other families. This is an Egyptian family that's homozygous for a mutation. This, interestingly, is a family from Labrador and Newfoundland. It's an Irish population that has one mutation. It's a 14 base pair deletion here that frame shifts. And then two others that have mutations out in this region. So that's the current state of where we are. And Wnt1, we don't know how it works yet. We have really no idea. There are all kinds of, it's a signaling molecule. When we saw it come up, we were clear that that was it because of what Matt had done previously, showing that one of the proteins it binds to, LRP5, when it has mutations in it, can give rise to dense and light bone disease. So this is the clinical picture. So here's this is the OI classification of almost all the people that fit into all of these other categories would fit into one of these, more or less. There's a lot of controversy now about how do you classify, do you call them by different numbers, and that's up for grabs at this point. And so in this pathway, we've begun to identify mutations that are here, so type 1 collagen gene mutations, and then mutations that affect all of the proteins, or many of the proteins that are involved. There still are plenty of samples left. We just don't know where they're going to be. But you can see here, this is the distribution of relative mutations in these genes that we have from our lab. You can see about 4.5% versus 95.5% in the ones that affect type 1 collagen. There are also recessive COL1A2 genes that are present. So it's a very interesting place to go. The people who have done the work are here. We appreciate the funding from the OI Foundation and from some other sources. And it's a growth industry is actually, it should be a growth industry because we should be able to do the same kinds of things that Michael has done and try and see whether or not we have ways to influence growth and development in these kids. Thanks.
Okay. Thanks very much. Well, we always believe in family advocacy, but I hadn't thought of it in terms of cousins. But
you never thought of that. I'm not sure I really believe you.
Anyway, we have time for just one question.
Yeah.
Yeah.
From a clinical perspective, if you have a patient in whom you suspect OI, autosomal recessive OI, would you do whole exome sequencing in the beginning to identify the underlying mutation?
So if you think that it's OI, that it's recessive on the basis of family structure and things, what we actually do is sequence all the recessive genes at the same time.
So you have a chip or?
Yeah.
Ah, okay. So you don't do whole exome sequencing, but you have an OI chip.
Yeah, so we do targeted sequencing. We actually have a plate. We're not up to chips yet, but there'll be next generation sequencing shortly for all of that. But it's now just it takes a week and a half.
Okay, Peter, thanks very much.
Yep.
Okay, we're going to move along here. And from Indiana University, home of the world's best basketball teams, is Kenneth Whyte. He's going to talk about FGF23 and a common denominator in metabolic bone diseases.
I first want to say thank you, Beyond the Institute, for the invitation to be here today. I get to talk about my favorite gene in one of my favorite places in the country. So it's a great match. Couldn't get any better. So fibroblast growth factor 23, or FGF23, is produced in osteoblasts and osteocytes. It is an endocrine factor in the kidney to reduce phosphate reabsorption. And the main reason that it's sort of allowed to be an endocrine factor is that it has a unique heparin binding site, and that it allows it to escape the extracellular matrix, unlike other FGFs that are trapped in the matrix, and they work as autocrine and paracrine factors. FGF23 also suppresses the renal synthesis of 1,25-dihydroxyvitamin D, the active form of the vitamin. It may reduce PTH production, and it acts through its co-receptor alpha-klotho. So FGF23 plays an important role in kidney axis. When there's an increase in serum phosphate or 125D, this drives FGF23 production. This leads to an increase in circulating FGF23, which travels to the kidney, and it suppresses the sodium phosphate cotransporters, as well as reduces the 1-alpha hydroxylase and increases the catabolic 24-hydroxylase. And the result of this is a paradoxical suppression of phosphate in the serum as well as 125D. The normal response would be in hypophosphatemia for vitamin D to go up. So when you're perusing the literature or looking at patients, if you have a decrease in both of these or normal 125D, I think it's a good bet that FGF23 could be involved in those diseases. So you might want to take a careful look. And then these suppress FGF23 production in making the feedback loop. So today I was asked to cover three diseases: autosomal dominant osteoglyphonic dysplasia, or OGD, and tumor-induced osteomalacia. And what they all have in common is that through three different mechanisms, it looks like at this point, serum FGF23 is elevated. So starting with ADHR, patients, like all the patients with high FGF23, have hypophosphatemia with normal calcemia. This is secondary to reduced renal reabsorption of phosphate. Inappropriately normal or low 125D, rickets osteomalacia, as you can see here in this subject. What's different about ADHR is that it can have completely unaffected carriers in terms of diseases that involve FGF23. There are essentially two types of these patients. There's early onset patients. These are very similar to the X-linked form of hypophosphatemia, where they developed lower extremity deformities. There's also the late onset, which is similar to tumor-induced osteomalacia. So they get the disease after the skeleton is formed, so they tend to have straight limbs, but then develop fractures, pseudofractures, but no bowing in the legs. So when I was in Mike Econ's lab as a fellow to walk down chromosome 12 as part of the ADHR consortium with Tim Strom, and we identified mutations in FGF23 that caused ADHR. And these mutations occur in a very narrow region of FGF23. And this is a subtolysin-like proprotein convertase site, or SPC, proteolytic site. And this site is composed of arginine, anything, arginine serine. So the mutations now are for ADHR have been found in arginine 176 and arginine 179 with glutamine or tryptophan substitutions. And these have really profound effects on the protein. These are Western blots of FGF23. In vitro, there's the wild type form. FGF23 full length is about 32 kilodaltons. So when the RXXR sequence is present, it can be readily degraded. And at the time, we didn't know why the RXXR sequence was important. We just knew it was really a pain in the neck to purify enough to do anything with. If you introduce one of the ADHR mutations, it tremendously stabilizes the form, the intact form, which is the bioactive form of FGF23. And what I'm going to point to in a little while is maybe why this RXXR site is emerging as truly a critical piece of the phosphate metabolism story. So as I mentioned, there are some differences with ADHR versus the other disorders involving FGF23. And this is a case that was characterized by Mike Ekrans and Paul McHenry. So this patient was documented to have the R176Q FGF23 mutation. At 3 months, she had normal phosphate. After puberty or through puberty, she had some back pain, hypophosphatemia, which improved with treatment, 19.5 years, still hypophosphatemic. And then at 22 years, full-blown ADHR with fatigue, ankle pain, osteomalacia. And you can see her bone scan here with pseudofractures in the legs, fractures in the ribs. This also happens to male patients. This is a documented R176W mutation by Greba et al. 27 years old, the proband of the family had marked hypophosphatemia and increased alkaline phosphatase, really no documented chronic disease in childhood. They probably had some, though, because it's said that he had slight bowing. And his father had no limb defects, but he developed really late onset ADHR and had hypophosphatemia over a 2-year period. So it's sort of this waxing and waning of disease. And this was really nicely characterized by Eric Immel on his work. So the top is serum phosphate levels, and the bottom is intact or bioactive FGF23. The normal mean is shown by the dotted lines. So the patients, when they're seen, are hypophosphatemic. Each line is a one patient that's being followed over time. Early in the disease course, FGF23 goes well up above the normal levels, and then on its own fixes itself. And this correlates with a rise in serum FGF23 levels. So actually, this open arrow is the space where Eric stopped treating this patient because she returned to the normal level on her own. There was no point in giving her phosphate anymore. This patient had a dramatic increase in FGF23 over time, which dwindled on its own, and again, correlated with sort of improvements in phosphate. But this really creates an ADHR paradox. Why is FGF23 high when serum phosphate is low? Serum phosphate is a very strong suppressor of FGF23. So phosphate, as I mentioned in these patients, is low. Vitamin D is low. PTH is usually normal. So there's got to be something outside the normal phosphate, calcium, vitamin D homeostatic system that can regulate FGF23. So there were 2 mouse models available at the time. There was an FGF23 transgenic mouse. This is markedly hypophosphatemic, as you would expect. There's really no control of expression. FGF23 knockout mice, they were hyperphosphatemic. They died at 8 to 10 weeks of the hyperphosphatemia and calcifications. These are tremendously useful models for understanding FGF23, but we felt it didn't quite recapitulate the phenotypic spectrum we could see in ADHR patients. So we developed a knock-in ADHR mouse that carried this R176Q FGF23 mutation, and we picked this mutation because the largest family that we had had this change, so we thought it might be a good resource for phenotyping. So if you look at the bone RNA from those mice, here's the RXXR serine site. In mice, it actually takes 2 mutations instead of a single point mutation, just because the codon used in the animal is slightly different than the human. This is RNA, so you can see here's the wild type mRNA, and here's the mutant mRNA. If you look at arginine 179, they have both copies that are correct, showing that the mRNA at least expresses our mutation. So we looked this mouse up and down under basal conditions, and it had a slight but significant reduction in bone mineral content, and that was it. I mean, really no home run ADHR phenotype. I mean, this was, as you can imagine, incredibly disappointing after all that work to make a knock-in mouse. So we thought, maybe we ended up with the unlucky carrier phenotype, which still is unique to ADHR. We almost wrote a paper that said, there must be more to ADHR than an FGF23 point mutation. I mean, there may be gene-environment interactions. So our biggest question is, does the disease have to be induced? And at the time we were doing our studies, there's a lot in literature about iron metabolism and FGF23. And here's two examples here where FGF23 was inversely correlated with plasma ferritin, so these patients were slightly anemic. And also iron infusion for anemia elevated FGF23. And there's also some human data going on that I'll talk about after we talk about the mice. So if you look at the iron requirements at puberty, when ADHR can have onset, iron requirements are very high as the teenager is going through puberty, and with the terrific diet that teenagers often eat, you can see why they become anemic by not getting enough iron. If you look at pregnancy, this graph is from 1948. It probably hasn't changed much since then, but of course, pregnant women before childbirth and postpartum are known to have reduced serum iron concentration. So these are two situations where ADHR is onset, but it points to low iron as a factor in FGF23. So we basically took the ADHR mice. We had nothing to lose. Some on control diet and some on low iron diet. So this color coding is going to come through in a few graphs. So this is the wild type on the control diet. This dark blue. The light blue is wild type on the low iron diet. The orange bars are the ADHR mouse on the control diet, and the ADHR on the low diet is the light orange bar. So we looked at the serum biochemistries. They were hypophosphatemic, only the ADHRs on the low diet. Calcium, again, normal. Alkaline phosphatase was up. PTH, there was a trend. It was not significant, but you can see the trending towards up. So basically, by putting the mice on a low iron diet, we could recapitulate the ADHR phenotype in these animals. There's two ways to measure FGF23 in rodents. There's an intact ELISA, where the capture and the detection antibodies are on either side of this RXXR motif. There's also what's called the C-terminal assay, where the capture and detection antibodies are on C-terminal side of this RXXR site. So what you get is full-length FGF23 in this ELISA, as well as all the fragments that are made from cutting this at this RXXR site. And I should mention that the intact is thought to mention the bioactive form of FGF23 because they're conformation-specific antibodies. So they detect FGF23 when it's properly folded. So we looked at FGF23 at 6 weeks in these animals. Again, here are the same codings. Only the AHR on the low iron diet had a significant proportion of these animals had elevated FGF23. Wild type control and low and ADHR control were all not different from each other. If you look at the C-terminal assay, I'll start with the ADHR low iron mouse. So this is, the C-terminal assay is increased, which fits with the intact assay because they do measure some of the same things. So that was fine. What we didn't understand was that the C-terminal assay was up in the wild type mouse on the low iron diet, where the intact was absolutely normal and phosphate was absolutely normal. At 8 weeks, the mice had inappropriately normal FGF23 and low 12. All the other conditions were absolutely the same. All the mice on low iron diet had very high C-terminal FGF23 concentrations. Phosphate was normal at these time points, and again, intact was normal. So we wondered, really what's going on here? We looked in the bones of these animals. These are immunoprecipitations from lysate. This is in body control. Wild type and ADHR animals appeared to have similar normal FGF23 levels from bone. And when you place them on low iron, FGF23 protein really went through. I mean, there's all different pieces and parts here, but you can perceive that there's a lot more protein there. So what was driving the protein? We looked at the RNA. Wild type and ADHR control animals on the diet, pretty normal, pretty similar mRNA. You put them on the low iron diet, FGF23 mRNA absolutely goes through the roof. So low iron is an incredibly strong driver of FGF23 mRNA. So at the genomic level, FGF23 is being controlled by iron. We looked at the bone phenotype, again, wild type, control low, pretty nice margins, they looked fine. Look at the ADHR animals, they had marked osteomalacia, you can see they're wider bones, they had slightly larger growth plate, consistent with the ADHR phenotype. They also had osteocytic lesions, which we thought was pretty interesting. This is seen in some X-linked hypophosphatemic patients, but there's really, I don't think the human biopsies are available anymore. And so FGF23 is made in osteocytes, so we don't know if anemia causes some defect in the local regulation of FGF23 in osteocytes either. We looked at the renal effects. FGF23 is known to suppress the sodium phosphate cotransporter in the apical membrane of the brush border and proximal tubule cell. Again, control diet, similar amounts of NPT2A. You make the ADHR animals anemic, and sure enough, NPT2A disappears because FGF23 is high in these animals. We looked at the metabolic enzymes for 125D. Alpha hydroxylase was suppressed, just like injecting FGF23. The 24-hydroxylase was elevated, and vitamin D in the serum was also low, similar to the ADHR phenotype. We wanted to know if the circulating FGF23 had effects in bioactivity. So FGF23, as I mentioned earlier, partners with klotho and an FGF receptor to signal, and this occurs through phospho-ERK signaling. So you can basically just map phospho-ERK in the kidney along with klotho to see where it's signaling. The green indicates klotho across the three conditions that are normal, and phospho-ERK signaling was only found localized with klotho in the ADHR mice, indicating that this elevated FGF23 really did have biological consequences in vivo. Eric Immel and Mikey was in parallel performing human studies on FGF23 and iron. Eric looked at a population of ADHR patients and found that the patients with lower serum iron had higher intact FGF23 and C-terminal FGF23. Again, this is okay because they measure some of the same things. He looked at control individuals. Only the C-terminal FGF23 had correlation with low iron, just like in the wild type animals. C-terminal is very high under low iron. In control individuals, the intact FGF23 was not correlated with iron. And we think this is because normal individuals can control the level of intact FGF23 by cutting it apart, so that phosphate, vitamin D, PTH, all the things we think of that normally regulate FGF23 are regulating it in these people. Eric also looked at some longitudinal samples he had. This is phosphate on the left and 125D on the right. Low iron resulted in hypotemia, and low iron also resulted in low 125D. So low iron, we think, drives FGF23, which reduces phosphate and vitamin D over time in these patients. So it seems to come full circle, and the human data reflects, what we found in the mice, or vice versa, however you want to look at it. We also looked at the regulation of FGF23 in vitro. Deferoxamine is a clinical iron chelator, so we used UMR 106 osteoblastic type cells that express FGF23. If you remove iron by adding DFO at 25 and 50 micromolar, you get increased FGF23 mRNA. This is the positive control, which is a transferrin receptor, which is known to be upregulated by anemia. This could be blunted by inhibiting MAP phospho-ERK with a MAP kinase inhibitor, UO126. We also used L-mimazine, which is a mimetic in the iron-sensing oxygen pathways. If you add L-mim to the UMR cells, you can also increase FGF23, showing that not only does this occur in vivo, but this is a local effect, most likely on bone itself. So this is a summary of where we are for ADHR. We believe that interactions cause late onset ADHR. In wild type individuals, there's an iron deficiency, which if you have an iron deficiency, it raises FGF23 mRNA. You carry the arginine at position 176. This allows FGF23 to be regulated in kind of a new secondary step that we never really appreciated existed at all. So you have this kind of mix of intact and C-terminal fragments where this proteolysis step, I think, was underappreciated by most of us in the field, realizing that if you do have a challenge, you may want to keep phosphate so you can control it at another level. So how all this happens is really an open book. In ADHR, iron deficiency stimulates the FGF23 mRNA, but you carry this mutation, it's now more stable, and then you get elevated FGF23 when you're driving mRNA at a high level. The next disorder is osteoglyphonic dysplasia, and this is a family that we looked at. They have mid-phase hypoplasia, shortened digits. They have craniosynostosis. And you can see here the baby picture and the son from the dad. OGD is also associated with dwarfism, which is a bit strange for disorders involving craniosynostosis. It's usually skull or bone. And I mean, I have to come clean a little bit because this guy is actually an NBA basketball player. So he's not quite this short. I think that we looked him up. I think he's about 6'9". So, you know, he's short, but I mean, still, I mean, mental capacity was good. He was actually a starter at a drag strip, you can see here. So a lot of these patients could lead normal lives for a while. This disease can be severe. It can lead to osteoporosis and cytic lesions, and you can see the lesions here, very low DEXA analysis. And the biochemical phenotype was really what turned us on to these families and interested. So if you look at serum phosphate, it was low. Calcium was normal. 125D was low. So low phosphate, low vitamin D points again to FGF23. So we sequenced these families. They're old school sequencing. There's the control individual, normal sequence. The grandmother was okay. The father had this missense mutation, a tyrosine to a cysteine, and so did the son. And this is in FGF receptor 1. And to also come clean is that Klotho wasn't known at the time. So we were sort of sequencing everybody we could for these FGF receptor mutations to try and identify the FGF receptor that bound to Klotho. Or I mean, that bound to FGF23, because Klotho wasn't known. So that was the point of doing all these studies. So the mutation occurs just upstream of the transmembrane domain. This is the FGF receptor kind of tilted on its side. Here's the transmembrane domain and the intracellular tyrosine kinase domains. Here's the transmembrane domain boxed, and the mutation, it's conserved, that tyrosine is conserved in mouse. And also, it's known that mutations in FGFR2 at exactly the same spot cause Bayer-Stevenson-Cutis gyrata syndrome. FGFR3 in the same spot causes a very severe disorder, thanatophoric dysplasia type 1. So it was consistent with other mutations that are known in the FGFRs. Now, this patient really put it into perspective for us for FGF23. We called this patient 3. Again, she had similar phenotype, really severe lytic lesions in bone. What was different about this patient was that she had an asparagine to isoleucine mutant N3, but had elevated FGF23. So it looked like these receptor mutations were leading to elevated FGF23, and that's why they were having the phenotype. If the receptor was the receptor and it was an activating mutation, we would normally expect FGF23 to be suppressed because they're hypophosphatemic, but that's not the case. So it looks like more the receptor drives FGF23 production. FGF23 then gives you a phenotype. And unfortunately, this is actually a de novo mutation that neither parents had in their blood lymphocytes. So the patient was really bad luck. So the FGF receptors work by forming heteromeric FGF, a receptor subunit, and heparin. We hypothesized that these cysteines, just like the other mutations like achondroplastic dysplasia, would free up cysteines, bring the receptors together and signal. So we collaborated with David Ornitz, who ran FGF receptor assays and osteocalcin promoter luciferase. So even without adding any FGF2, at the basal state, the receptor was 10 times more potent at signaling without even ligand there. So this is a ligand-independent mutation. And by adding FGF2, it was more significant at every concentration. So this is a summary of these 3 mutations now that have been found for osteoglyphonic dysplasia. They're in this small area surrounding the transmembrane domain and highly conserved residues. The cysteine mutations make sense to us because they free up a dimer for signaling, but the N330I, we still don't have a handle on why this causes such severe disease. And it looks like the further N-terminal you are in genotype, the more severe your phenotype. All of these patients died in childhood from their disease, where these patients did fairly well for a while, but they had an FGF23 phenotype. These guys had an FGF23 phenotype. The patients that I didn't show here were fairly normal. They have slight lytic lesions. They're all short, but really no phosphate or FGF23 defects at all. So there does seem to be, at least with like the 8 people we have, some genotype-phenotype correlation. The last disorder that I'll go through kind of quickly is tumor-induced osteomalacia. This is a disorder of severe hypophosphatemia. And this is associated with tumors often originating from mesenchymal origin, and you can see it here in the patient's knee. So TIO, for a long time, has introduced the idea of a phosphatoning factor that regulates phosphate because they knew that if you found the tumor and took it out, the patients were absolutely fine. And they had hypophosphatemia, inappropriately normal vitamin D, again, all things that sound like FGF23. So shortly after we found FGF23, we said, we have a great phosphatoning candidate. So we collaborated with Michael Manstadt and Harold Eubner at Mass General, and they sent us a northern blot that had RNA from 5 tumors on it, as well as control tissues, and we blotted it with FGF23 cDNA, and it was as hot as could be. You know, some northern blots stay in the freezer for like 2 weeks. This is a 30-minute exposure. I mean, it's so hot, it's unbelievable. And I was worried. I went and told Mike, I'm like, you know, Harold's going to kill me. I've ruined like the only one of these that exists in the world. But it worked out for the best. It was fine. We had an antibody FGF23, and it was present in the tumor as well. At the same time, Kieran Pharmaceutics was doing really nice work. They did a subtractive approach and found FGF23 only in the tumor, not in the adjacent bone tissue. They made Chinese hamster ovary cell lines, injected them into nude mice, and they basically got the ADHR phenotype with widened growth plates, low bone mass, and for the first time made the connection between reducing NPT2A and 1-alpha hydroxylase and showing that FGF23 had control over both of these genes. And now TIO now has its own tumor classification. It's Pituitary mesenchymal tumor mixed connective tissue variant tumor, a PMT-MCT. Can have spindly shaped cells, and it can kind of have a smudgy matrix, can be highly vascularized, and can have osteoclast invasion. We did some work with Leanne Ward, who had a tumor, had a patient with a tumor in their wrist. Once it was removed, you can appreciate the difference in bone mineral density here in this 9-year-old girl. Here's her severe osteomalacia and smudgy calcium. Goes away after you take out the tumor. Harold Eubner did some really beautiful work, a panel of patients that had OOM. This is just oncogenic osteomalacia, another name for TIO. So before surgery and after surgery, measured FGF23, and one of the first ELISAs was made, and FGF23 falls almost into the normal range, as you can see here, after surgery, and I won't discuss XLH because that's up next. These TIO tumors, though, can be very difficult to find. People have lived forward with these, just take phosphate and vitamin D therapy. They rarely metastasize, although there have been deaths due to this. And standard MRI can be hard to find the tumor, especially when there may be multiple lesions. We did some work with Dorothy Schulman and showed that recall imaging from MR using gradient echo actually reverses the image so that it can be light against a dark background, may help you find these tumors, especially when there's multiple. And actually, this tumor was taken out the first time and found not to have FGF23, and the patient didn't get better. This tumor was taken out. We looked at it for FGF23, and sure enough, it produced plenty of FGF23, and they took it out, and the patient was cured. There's also systemic surveys to find FGF23, and it's done by venous sampling. Like again, here's another case where you have several lesions in the person. It's a thigh lesion. They did venous sampling in this JCM paper, and right next to the tumor where all the other values are around 70 or 80, on the way back in the venous system, right around the tumor, you get high FGF23 levels. So this may provide another way to go about finding these tumors when they're very, very difficult to find. So to summarize, this is, I called it a unifying model, but I mean, the only thing that's unifying about it is FGF23 is high that we know of. So in ADHR with a fall in iron, this stimulates FGF23 mRNA in bone. These patients carry the R176Q mutation, which raises intact FGF23 and leads to renal phosphate wasting and the disease onset you get. FGFR1 mutations somehow also raise FGF23, whether it's at the level of the gene or somehow interfering with processing. Gives you the same phenotype, although because it's activating and expressed in bone, all the patients are also dwarfs. And in PMT, MCT tumors, there must be some transformation event that starts these benign tumors forming. They definitely have high FGF23 mRNA, but they overproduce wild-type FGF23. So again, whether there's a cutting defect here or it's just a matter of they're producing so much that the cells kick it out intact, it's really not known, and gives you the same disease phenotype. So I'd like to thank the members of my lab. Emily Farrow and CJU did most of the work, as well as our clinical collaborators and advisors. Jim Fleet helped us with the diets. Immutopics gave us all the first generation C-terminal mouse kits, and Leanne Ward for being a great TIO collaborator, and all the investigators involved in the osteoglyphonic story and our funding sources. And thank you for your attention. Appreciate it.
We're running a little bit behind time, so we'll just take one question and then we'll move on. Michael?
The iron story translate over to XLH and virtually all the disorders of high or low FGF23?
Yeah, I mean, people are starting to look now. Maybe Tom can address that better than I can, but the patients don't seem to have iron trouble. It seems to be a one-way thing where anemia causes this at the right time, and maybe it sets them off where they can't recover. But as far as I know, some of the other disorders don't involve anemia. Yeah, that's a good question. I mean, people have looked at intact FGF23, but we're all wild type. So what really needs to be done is the C-terminal assay run in that case.
Okay, thanks very much. The next talk is going to be by Thomas Carpenter. And since it's 2013, he's going to bring us up to date on the X-linked hypophosphatemia clinical update and prototype renal phosphate wasting disorder.
Use the mouse as your pointer, but you can actually go through slides with it. Is this pointer on here right there?
It is. It's pretty dull.
Okay. Thanks, Jose, for inviting me. This has been a great day. I have certainly enjoyed a break from the blizzard in the Northeast, and I have enjoyed the conference even more. It's been a lot of fun and a lot of good stuff. I'm going to spend, in a way, a complimentary talk to Ken's, talking about the prototype disorder that's more common than many of the other FGF23 related disorders, and that is XLH. And should point out that
should change my disclosure slide from Inobia to Alexion, as I do serve on one of their DSMBs, and I also work with in some of the protocols that have been used to develop therapies for XLH. So we've heard today about phosphate. It's the element phosphorus with a complex of 4 oxygen atoms around it. It's the stable form of phosphorus in the body. It's necessary for the formation of hydroxyapatite, which is necessary for making a stable and fun. So I want to spend the next 20 minutes or so on what is life with XLH like? What is the natural history? What do people that have this disorder live with? We'll also talk a little bit about why these things happen. What is the pathophysiology of the disorder? We'll move along to what we currently do to treat this disorder and what we need to fix about treatment, and also point out some new treatment targets. So XLH occurs across a very wide spectrum of disease. X-rays from 2 women in their 60s. Whoops. On this lady has a virtually normal skeleton. Her disease has been manifest primarily by dental problems with the disease. This is a woman about 2 or 3 years older who also has really significant, and has had her whole life, very significant skeletal disease. Here you see a dramatic bow osteophytes forming off the tib and fib, abnormal metaphyses, sort of classic findings of this disorder. Long bone changes occur early. They occur as early, if you aren't looking for them, noticed often by the grandmother at about age 1.5 or 2, somebody that doesn't see the child every day. These are the sort of classic X-rays that somebody might show up in childhood with for the first time. Another thing that happens is tibial torsion. It becomes clinically evident a little bit later than the bow usually, and it leads some children who actually have reasonable leg alignment in terms of a bow defect to require a derotational osteotomy, as this little girl had to have, to twist the lower part of the lower extremities back out so they're not extremely pigeon-toed. Radiographic rickets usually appears in the same time frame if one looks. Other long bone changes include the failure to grow. This is from a recent paper that actually looks at a variety of children and growth charts and the sort of early fall off in growth that's very typical of this disorder. This translates to a therapeutic point in that it seems that the height outcomes of children that have therapy applied early, before age 3 to 4 years of age, do much better than those in terms of final adult height than those that are treated later or have their initiation of treatment later. Another thing we see is some changes in the geometry of the skeleton, and in this particular case, this very significant coxa vara, which to compensate for requires often a very lordotic posture. I don't think that this is really a phosphate-related complication of the disorder, and it's something we'll mention in a minute, and that is this disorder appears to be something more than a mineral deficiency, and in fact might be a hybrid disorder between a mineral deficiency and perhaps a skeletal dysplasia-like syndrome. Another probably non-phosphate related disorder is the Chiari-1 malformations that these children get. This cerebellar tonsils are low here. She actually had craniosynostosis and had enlarged ventricles and had to have a shunt placed. This is actually after some decompression of the ventricles. Frontal bossing is typical, even without the severe cranial abnormalities resulting in needing a shunt. The skull shape can be somewhat different. So these are all things that are occurring very early on and may relate to extra-renal effects of growth factors. And of course, the classic osteomalacia is part of the skin. See that suggested by the ground glass appearance in this child's X-ray. And if one were to biopsy that, this is actually from an adult XLH patient, the accumulation of the unmineralized osteoid showing up in red here is excessive and creates the flexibility of the bones that tends to be compromised with weight bearing and therefore bow even in adulthood. Another major problem that folks with XLH have to live with are dental problems. The typical dental problem that is experienced is a dental abscess, as shown here. The tooth, the major abnormality in the tooth is not so much in the enamel layer, the hard exterior shell, but in the secondary dentin layer, which in this EM here is shown from a tooth from a patient with XLH and in a control person. And the dentin, which starts here, is there's a very thin stripe of what's called predentin, a mantle of predentin that's normal. But then as you get into the dentin layer, it's got these poorly coalescing calcospheres that relate in a porosity that are thought to break down the anatomic barrier that allows oral flora and microorganisms to get into the pulp of the tooth. And what you see then is after organisms can get in here, there's around the root of the tooth inflammation related to this infection. Oftentimes, by the time these are diagnosed, people can't grow organisms out, but this expansion of space here represents the inflammation and subsequent tissue reaction where inflammatory cells have come in and created the space between the bone and the tooth. So the normal childhood course, with or without therapy, often results in a variable progression of the bow deformity. Sometimes even in our most compliant patients, there's a requirement for orthopedic intervention. Patients, when they're a little bit older and start doing more things, usually what are our current approaches to therapy? We've understood this as a disorder of phosphate wasting primarily, and so we phosphate, and because of the generated amounts of 125D, we replace with 125D, a classical endocrine therapy to an endocrine deficiency disease. But we run into problems with this therapy, and those problems are as such. Giving phosphorus actually compounds the tendency to secrete PTH in this disorder, so parathyroid hormone levels can get quite high. They need to be monitored, and if not controlled, they can create further skeletal and phosphate wasting problems. It also turns out adding the phosphate increases FGF23, and it turns out that calcitriol also increases FGF23. So now we've compounded our protect serum phosphate by giving agents that, in fact, make phosphate wasting worse. And one can see this. This is a study from our hospital research unit where we've taken a group of patients that have been asked to withhold their therapy for a couple of weeks, and those that came in on therapy. And the FGF23 levels are higher in the treated group. The 125D levels, paradoxically, are actually higher in the group not getting 125D. The phosphate levels are actually higher in the group not getting phosphorus, and this is because their TMPs aren't as suppressed. So there are other complications of this disorder that become major problems after a pediatrician like myself deals with them, and I talk a lot with our adult group that I graduate these children to, and they have a whole different set of disorders that have, until recently, been largely neglected in terms of therapy. I have mentioned the hyperparathyroidism. Many patients have gone on to parathyroidectomy, have also been treated with the newer hyperpara drugs such as cinacalcet. Osteophytes, arthritis is very common. Osteophytes and calcified entheses can be quite painful and quite limiting as well. This is a 50-year-old man who has developed bone spurs all over the place. You can see his Achilles tendon is beginning to calcify. His mobility is highly impaired by these changes. He also has significant bone spurs coming off of his femur. He had significant disease as a child, you can see from the rods. Here his patella has fused to the distal femur, and the quadriceps tendon is beginning to calcify. All of these result in tremendous limitations in his range of motion. This is also data from our CRC from some years ago showing in adults, these are control adults, these are untreated adults, and these are treated adults, and this red line is the normal level in this assay for parathyroid hormone, and you can see both in treated children, in treated and in untreated adults, hyperpara occurs, and it seems to predominate overnight. So what are some new strategies we can look at in improving this therapeutic problem we have in XLH? One consideration that has been raised in the past has been to provide, go right where the money is. The mutation is in FEX, provide FEX. FGF23 is elevated, so one can maneuver a number of things here, such as suppress FGF23 synthesis, inhibit its action, try to block the receptor, or try to block the effect of clotho. And we'll talk a little bit about a recent publication that enhances the breakdown of FGF23. PTH, as we've mentioned, can be elevated. There are some studies that suggest it can potentiate the action of FGF23, so suppression of PTH is probably a very good thing in this disorder. And there's a whole another axis that I don't have time to go into great detail about, but have alluded to, and that is that sibling proteins have these ASARM fragments that are thought to regulate mineralization itself, and in some cases, at least experimentally, modulate phosphate wasting. That's not been as clear in vivo but another whole strategy of targets to hit may be in the sibling protein realm. This is a study lent to me by Inobia some years ago that shows their data in the XLH mouse, or the hip mouse, giving for quite a long time soluble FEX. And this is a bunch of control mice at 29, 57, and 92 days out of receiving this replacement enzyme. And these are hip mice and treated hip mice, untreated and treated. And you see there's no change in the serum phosphorus. There's no change here in the bone lesions, and there's no change in the growth plate thickness. These being the controls and these being the hype and the treated up here. So that didn't work. So another thought that we should discuss is manipulating FGF23. I think you saw a slide just like this from Dr. Whyte, demonstrating that there's specific cleavage sites, and the cleavage renders the intact molecule into two inactive fragments. But researchers at NYU have identified that this C-terminus can be exploited to inhibit the receptor complex as it binds to the receptor. And in their study from now a couple of years ago, they infuse to rats here either the intact molecule or the C-terminus, and you can see that intact FGF23 does what you would expect, drops the serum phosphorus level. However, the C-terminus raises the serum phosphorus level, and this is explained by the effects on free excretion of phosphate, whereas the intact molecule increases phosphaturia and the C-terminus reduces phosphaturia. A potential approach. Our group, and studies done a couple of years ago, identified that calcitonin, after a single injection of calcitonin, we could see in XLH patients about a 20 to 25% drop in FGF23 that results in a transient rise in the serum phosphorus level. And so we're currently doing a intranasal calcitonin study over a 2 or 3 month period to see if this effect can be sustained. Again, the FGF23 molecule in a recent paper that comes from the Dresner laboratory, in which the complex that results in cleavage of FGF23 might be a target by which one could affect the accumulation of the active molecule in this process if one can stimulate this 7B2PC2 complex. He has identified that one of the cofactors for this convertase is underexpressed in hype. The brown staining material here is underexpressed in hype bone compared to normal bone, this being the quantitation, and suggesting that the reason that patients with XLH have high FGF23 levels is because there's less of the active form of this convertase around. So his theory is activate these and will reduce the levels. It was identified that a string of 6 D-arginine molecules can do that. And his study in mice, I guess we'll start here. These are the hype mice. These are the hype mice that have received hexaarginine. Their serum phosphates on this axis go up. And there's really not much difference between the wild types receiving vehicle or receiving the hexa-D-arginine. This was accompanied by a drop in FGF23 levels and FGF expression, as well as circulating intact FGF23. The study was very nice at the histological level, showing, again, after hexa-D-arginine, the smudge of mineralization going away into nicely defined stripes of orderly mineralization a week apart here, and reduction in the unmineralized osteoid in the treated height bone compared to the untreated. The bones here, this being the height bone, this being the normal, the geometry of the bone is much more normal than in the untreated state. Other approaches, I think everyone has heard the story about a neutralizing antibody to FGF23. These are mouse studies that were done by Kieran several years ago. A similar story in a dose-dependent way, showing nicer femurs and tibia, less osteomalacia. There's restoration using this antibody of bone volume, growth weight, mineral ash weight, and the serum phosphorus levels. This is now in human trials. First single dose study has been completed and the manuscript's in preparation. The multi-dose study is ongoing, and there's looking towards designing studies in children is the current activity of that company right now. What about adjunctive therapy? People have talked about using growth hormone. All extensive reviews have been limited in terms of how much gain there is for the pain of that approach. Many patients require surgery. I mentioned most of these are for correcting bow defects, but some are done to correct this tibial torsion that occurs. Guided growth using staples across the growth plate have also been very helpful. I want to stop here with just recognizing the tremendous for people that are looking for support that the XLH network does, and that's their website. They do a number of things. One of the things they did in our region last year was help support, I guess for the second year in a row, XLH Day, where the kids get to pick on their doctor who has been nagging them all year to take their phosphate. It wasn't too cold that day, but I did get wet quite a bit. Those are our folks that help out, and I want to thank the NIH for getting us launched in terms of a center via their P50 mechanisms, our own Yale Center for Clinical Investigation, our studies on, and I'm going to stop right there.